Let's extract 1 million reads from read 1 and read 2. One reason to reduce the data is simply to reduce runtime for this lecture. You can do this by using seq-hit command. Copy this command and paste it. This will extract 500,000 reads from read 1 and will generate a new file named with .1million.fastq. Then run the same command for read 2. Okay, let's check the result using seq-hit command. Now the results are shown here. The original data contains 1,500 million base pairs, which corresponds to 600-fold coverage of the genome size. The extracted data contains 250 million base pairs, which corresponds to 100-fold coverage of the genome size. In general, 100-fold coverage is enough for bacterial genome assembly. This is another reason to reduce the data. Coverage or depth of coverage represents how many times a genome has been sequenced. In this example, the genome has been sequenced 5 times. So depth of coverage of this example is 5. Coverage can be calculated using this equation. The total read number is 1 million and read length is 250. And the genome size is 2.5 megabase pair, so it is estimated as 100-fold coverage. Next, let's check the quality of read data using fastqc. Run this command, fastqc, and the file name is .1million.fastq. fastqc will generate a result in an HTML format. This is the result generated by fastqc. Let's download this file and open this with your web browser. Download this and open. The overall quality of the data is quite good, except 2 warnings. One is for sequence quality. The quality of the 3' end is a little bit low. It is no problem, as this trend is very common. Another warning is about adapter content. Here, you can observe adapter sequences near the 3' end of the read. Adapter is a sequence attached to each end of the DNA insert. When the insert length is short, the adapter sequence may appear at the 3' end of the read sequence, like this. There are many tools to remove adapter sequences. We will use fastp in this lecture. Fastp can be run using this command. Copy this command and paste it. Here, output files are named with .1million.fastp.fastq. After that, let's run seq-hit and fastqc again. In this lecture, I executed fastqc in advance. This shows its result. Now we can see that adapted sequences have been removed successfully. There is a new warning about sequence length distribution. This is okay because we have trimmed the reads and various lengths of reads are included in the data.